Vinayak is also there. So today my topic, which is assigned to me, is advances in diagnosis of infectious diseases. Uh, so diagnostics actually, you see the infectious diseases, if we classify, uh, there are different etiology like bacterial etiology, viral etiology, fungal etiology, yeast is the etiology, and parasites are also there. In parasite also you can classify in a very different way. And if you just enlist the human and animal infections, so it will go near about 2000 infective agents are there which are causing disease either in humans or animals or to the both. And whenever we go for the diagnostics or diagnosing any particular disease condition, particularly in veterinary medicine, still our major approach or the classical approach is the detection of etiological organism, whatever it is. Maybe it's a bacteria, it's a virus or a fungi or a parasite or whatever it is. Our classical approach is to detect this etiological agent in the clinical sample which is received to us, or when we do the necropsy, we collect the samples and we try to isolate the organism with the something assumption in the mind that this could be the cause of the disease depending on the symptoms. And then the all isolation process, detection process are targeted or the moving on that direction only. And the other way where the isolation and detection of etiological agent is not possible, we try to detect the antibodies against this suspected disease in the sera. And then we confirm that, okay, this might be the disease in that. And so this is about the whenever there is some disease in animals is there, but many a times we perform the diagnostics or we perform the diagnostic test to confirm that the herd is free of some disease. And with this, now in this era, this is also very, very important. We have now globally eradicated rinderpest disease. Because why we say this, when we screen the samples, sera samples, we could not find any antibodies to it. Also, this issue is very, very important when we export some animal products from India to some other countries. If the importing country doesn't have that disease, they specifically need that material which is coming, they are derived from the animals which do not have that disease. In that condition also, we use some diagnostic test and then declare that it is free of the disease, and then it helps in the trading and export of some material, and it gives you some status. Globally, some status is achieved by that. So that's why also that is the reason also that we do some diagnostic test. So not only that some ailing patient is there and we collect the sample and then do the diagnostic test and confirm the disease, but diagnostics can be performed or diagnostics test can be performed just to declare that these herd, this area, animals or chickens or birds, they are free of this disease. It helps in the trading like that. So here the diagnostics are used. Now coming to the classical ways, though my topic is advances, I will take it on the latter part, but we will just fastly go through what is routinely done with the classical way. So for the bacterial diseases, when we are going to detect the etiological agent, either in clinical samples or necropsy, the traditional way is to, from the samples which we receive, we go for the bacterial isolation. We stick it on the agar plate, we do the gram staining, and then gram positive or gram negative, like that we classify them, and then do some biochemical test, do the INVIC test, and some required biochemical tests that can be also done. And then we come to know that, okay, this can be this organism which has caused the disease, So this is the traditional way, but it needs a lot of time to confirm. Also, the situation goes worse when the samples like tuberculosis comes. This tuberculous bacillus, they took a very long time to culture. They will grow, they will take near about 28 days to grow on the plates. So what can be done? We cannot hold the diagnosis for 28 days. So it is very difficult. Also, there are certain organisms like mycoplasma, they are very difficult to isolate even. Brucella is another example here. If you see the isolation percentage of the mycoplasma and Brucella is just 15%. From the all positive cases or the confirmed clinical case, only 15% you can get the isolates. So they are very difficult to isolate. Same is the case with the mycoplasma. Even in that case, it is lesser than the 15%. So these are few hurdles, particularly as far as the bacterial diseases are concerned, that with the routine techniques, we may not have some isolates after the routine techniques. Now coming to the viral etiology, the traditional way is to inoculate the embryonated eggs, or you can isolate the virus on the cell culture. So these two are the classical approaches for the detection of the etiological agent if virus is the etiology. So for this, we need very specified laboratories, because all laboratories cannot handle all the viruses at a time, because there are some specific laboratories like Pox lab will handle only Pox viruses. Morbillivirus laboratory will handle only morbillivirus, because for the viruses, different cell lines are required, and it is impossible to maintain all kinds of cell lines. Also, there can be cross contamination or like that. So there is a specified laboratories are required for the viral isolations and diagnosis of the viral diseases. Also, very expertise is also required for these individual viruses. Like now we have COVID also there. It is highly zoonotic. So some specific laboratories and expertise is also required to diagnose or to isolate these viruses. All labs cannot do it. All labs cannot do it. Also, there is a problem of the spillover of these viruses from the clinical samples, and it can get humans can get affected, or these other animals also can get affected. Maybe 20 years back, you might be knowing the epidemic of foot and mouth disease in UK. So that is the spillover of the virus from the laboratory. So there are many chances, if the laboratories are not well equipped, that there will be spillover and disease may spread. Then coming to the parasitic etiology, it is very easy to detect the parasitic egg in the feces. So best way for the parasitic etiology, what we do, for the gastrointestinal parasites, we sediment the fecal samples and see the presence of eggs are there. But if you are not very expert, all eggs look same. All eggs look same, and they cannot be differentiated. Even many a times, I have seen many persons in the laboratories, they could not differentiate whether this is egg or not. So these things are there. And as far as the parasites are concerned, these parasites, they have different stages in the life cycle. They have different stages. Also, they have multiple hosts. From one host, it is easy to diagnose some parasitic infection. But when it's of multiple hosts, then it becomes very difficult, because the intermediate stage is seen in some other animal, and a definitive host will have some other life cycle stage, and the lesions will also vary between the intermediate host and the definitive host. Also, few of the parasites, they form a cyst, like a neurocysticercosis. It will form a cyst which is a calcified, and in such cases it is very difficult to diagnose. Some parasites, they go and hide in the muscles, they go and hide in the bile duct, like that. So these are some issues, and the parasites which forms a cyst, you won't find any parasitic egg in the fecal sample. Then if the parasite is there in the muscle, you won't see any egg in the fecal sample. Also, if you go for the blood protozoan, those parasites, many of them, they look alike. Also, if you know that it's a pyroplasm, it becomes very difficult to identify it very correctly. So these are few hindrances in the correct diagnosis with the routine technique. These are best. These are very good. I don't say that we should not use them. They are very good. 80% of the cases we can very well diagnose with these routine techniques. But what about the 20% cases? So for these 20% cases, we need the advanced diagnostic test. And now we will see what are these advanced diagnostic tests. Maybe these diagnostic tests, they are evolved slowly. They have not come into the picture all of a sudden. So slowly they have been developed. So we will see how they have been developed and how they what are the recent tests that are available for the diagnosis of the infectious agent. So to overcome these issues, we have two approaches. One is the serological diagnosis. I have already earlier also mentioned about it. And another way is the molecular test. And this molecular test, if you see the 30 years back, the serological test were the like boom. Whatever the hindrances are there, that time we thought we can solve everything with the serological diagnosis. They are also very good. But as the molecular test, DNA-based or RNA-based tests have come, now they have taken upper hand over this serological. But still, I would consider this both way they are required for some particular diseases. These both we cannot compare them. They have their own importance, significance, and depending on the disease, depending on the pathogenesis of the disease, we can imply serological diagnosis for the detection of infectious disease or molecular test. We will see one by one what are these. So traditionally, how we learned about the serodiagnosis, all these tests we learned in our undergraduate or postgraduate teaching. Agglutination test, standard tube agglutination test, precipitation test, immunoelectrophoresis test, and latest is ELISA. Latest means it is 20-30 years back invention is there. And so first agglutination tests were developed, and we could diagnose many of the diseases with the agglutination test. Best example is salmonellosis, brucellosis, that we can go with this agglutination test. Then with the standard tube agglutination test, we can go for the titer of these antibodies. How much is the titer of antibodies in the sera, that we can know with this. Precipitation test is there, ELISA. Nowadays, almost ELISA has taken upper hand over the all other tests because it is easy, user-friendly. So that's why ELISA is used. So ELISA, when the ELISA first time it was discovered, it was a direct ELISA, and later on, as it was user-friendly, I would say that time, as compared to the complement fixation test and other very cumbersome tests, this was very easy. So there were a lot of research and development in this ELISA test, and then indirect ELISA was developed to know the antibody status. Then there was a question that, okay, we are detecting antibody. What about the antigen or infective agent? If you want to detect the infective agent in it, then sandwich ELISA was developed so that we can detect the actual pathogen in the clinical sample using sandwich ELISA. Then in veterinary science, we have to deal with the many species. Like if I tell you example of the brucellosis. Brucellosis, we can have disease in the buffaloes, we can have in the cattle, we can have in the sheep, we can have in the goat, we can have in the humans, like that. Many species are there. But if we do with the indirect ELISA, I think with that kit, we can go with the only one species disease diagnosis. But in brucellosis, as we have multiple hosts in this and disease in the multiple species, then the competitive ELISA was developed. So in competitive ELISA, you can test the antibodies of the almost all species and tell that whether that antibody is present or not in the clinical sample. So this is how it has been developed and very well used. ELISA. ELISA is still in many of the conditions or the situation, if you want to know the antibody, still first preference is ELISA. And now in ELISA, there are a lot of developments which I have not put on this picture. Earlier, when we used to do ELISA, we used to coat the plate today and conduct the testing tomorrow. So 2 days were used for the diagnosis. But now you get already pre-coated, then blocked, everything just to ready, prepare the plates are coming. You load the samples and you get the result within less than 2 hours. There are some certain kits which you can have the results within 2 hours. So this development has been taking place in the ELISA, which has made them very user friendly. Results are very fast, and you can give the diagnosis. So these are the developments which are happening, and with the use of peptides, particularly peptides in this ELISA, it has revolutionized this ELISA test. Revolutionized this ELISA test. The cost has come down, and all that has happened in this ELISA. So in 2012, there was one survey made about the ELISA, and those who are using this ELISA, what are the problems they faced, and how, as per the users, what is needed in the ELISA. So this is some ranking is this analysis is given here, and 8 years back, still there was in some tests there was a need in increasing the sensitivity. Still, if you see in the market, whatever the ELISAs are available, their sensitivity is between 80 to 90%. So that needs to go towards 98, 99%. That was the topmost questions or the opinion of the users were there. Then reducing the sample reagent, that is, we have very well taken care with the use of peptides and conjugating technologies that has been reduced. So these are different things. And the last one is the ability to miniaturize the format. That time it was thought, 8 years back, it was thought to be the least preference by the person, those who are doing ELISA. But today, if you ask, if you do the same survey today, the first thing will come is the ability to miniaturize the format. The duration for the ELISA should be least possible, maybe 2 hours, maximum 2 hours. When the sample is received, I have the plates already coated, block, everything. I have to add the samples, and within 2 hours, I should give the result. So this will be the first priority. So because of that, and miniaturization and the reducing time, the people, they have developed this reverse ELISA now, and this is very, very widely used and very nice technique, cheaper as compared to the plate ELISA, and it can be done at any place, and very good test, but it needs a lot of input technique, means intellectual inputs, as well as the precision inputs in the development of such tests. So with the routine ELISA, we need ELISA reader for that, and many things in that. In this reverse ELISA, we just need the cassette. We just need the cassette, and we have to put the sample here in the sample loading pad, and then you can get the result. C is the control line, T is the test line. If both lines are positive, the sample is positive. Now there are a lot development in this. Not only one disease, you can have 2-3 lines for the 2-3 different diseases. Like if there is a diarrhea, and I want to know whether it is a rota or a parvo or some other viruses, there is a development nowadays. You can have 2-3 lines for different diseases, and you can detect the infective agent. Also, you can detect the antibodies. Like in some of the diseases, you want to know whether it is IgM present or IgG present. So there are lateral flows developed where you can have 2 different lines, one line for IgM, one line for IgG, and one line for control. So depending, so as per need, there is a lot of development happening in this lateral flow. These are wonderful and very user friendly, point of care diagnostic, very well, and it is widely used also. In India, presently, there is an issue with this, that is the cost. So at present, many of the tests, they are imported, and they are bit expensive, not that expensive, bit expensive. So I think over the period of next 2 or 3 years, this cost will also come down, and this will be used everywhere. Then in the diagnostic of infectious agent, many of the times radioimmunoassay is not used, but it is used for the detection of the hormone levels in the blood. With this routine ELISA, there is a limit that we can go up to some level of the antigen which is present in the clinical material. But to increase the sensitivity of these techniques, the radioimmunoassays are used. Initially, they were for the hormonal levels, but now they have been also used for some of the infectious diseases. But now it is going on the back end because there's the risk associated with the radioimmunoassay, and you need various permissions to run this laboratory, and the rules are very stringent. So this has gone on the backside. And to overcome this, now there is a one optimizer is a technology which has come. What it is a digital detection technology with very high precision immunoassays there, and you can detect the antigen or the part of the infective agent at the femtogram level. Very sensitive, very sensitive. And this is improvement in this, that in ELISA, first thing, it is miniaturized also, and you can increase the sensitivity of the ELISA up to femtogram level. And this is one picture by using such technology, and by routine ELISA like HRPO conjugate ELISA, that you can detect the like 10 or 50 picogram per ml, and this plate capture ELISA, you can go up to 5 picogram. But with this optimizer, if you use this, and with this you can go up to 0.2 picogram or like that. I told you you can go up to femtogram. What is femtogram? It is 10 to the power minus 15 gram. Just think about it. So we know gram, milligram, macrogram, nanogram, picogram, and picogram earlier they used to call, but now it is a femtogram. It is 10 to the power minus 15 gram. So to this minute level, we can go and detect the antigen in the clinical samples. This is very important when the samples received is very less, or the antigen concentration in the sample is very less. Then we can use such tests to detect the antigen in the clinical sample. Then coming to another technology, that is latex agglutination test. So earlier I have told you that we used agglutination test for the detection of Brucella and Salmonella. It has not been developed for all other diseases. It is very good test. Farm side, on the farm you can do it. Patient bedside you can do it. On the beside the animal you can do it. But it has been developed only for 2-3 diseases, because this agglutination property should be there in the antigen to develop such test. But now with this latex bead can be conjugated to the protein which do not have agglutination property, and make it agglutinable, and then that can be used for the diagnostic test. So with the use of these latex beads, literally you can make any antigen which can form agglutination, and that can be used. So this is nowadays this technique is growing on, means many diseases it has been used, even for Anaplasma marginale also. And the beauty of this technique is that you don't need the infective agent as such to be used in the agglutination test, like we do it in the Brucella or Salmonella. You can have the recombinant protein of that infective agent tag it to the latex beads, and then use it for the agglutination test. So it is safe also, and it is cheaper. It becomes cheaper also by that way. So you can have the peptides, or you can have the recombinant protein, conjugate it with the latex beads and make it agglutinable, and that those tests can be developed. And this test can be done at the farm or beside the animal. That is the beauty of this. And with this latex, you can develop any for the any disease, you can develop the test for this, and this is very rapid also. Then in taking it forward for the proteomics, not only ELISA, but we can take the advantage of the some specific biomarkers for the diagnosis of the disease. And these are not new. These were also used since long time. And these include the technique like Western blot and immunohistochemical staining. Pathologists particularly take very pride of it, and mass spectrometry. By this way, you can diagnose many of the diseases. We will see one by one. Western blot. Western blot initially was used for the diagnosis of HIV. In our lab, we quite often use it for the diagnosis of the cysticercosis. Porcine samples, or even for the human samples, we often use it for the diagnosis of the cysticercosis, and that is the very good technique. Or I would say ELISA is also there, but this is more sensitive technique for the diagnosis of cysticercosis. Particularly in neurocysticercosis or other cysts, when calcification has happened, there the chances of release of antigen in the sera is quite less, and the concentration is very less. So ELISA can detect just antibodies, but if you want to detect the antigen, Western blot is the very good answer to it, and you can go for the by this way and detect the antigen in the clinical samples. Other thing that of proteomics that we can use for the identification of the infectious diseases, immunohistochemistry. So this is a picture of the this is from CDC website, and these are the rabies viruses that has been stained with the immunohistochemistry. So not only for the rabies, but in fact now you can use it for the any disease. You have to develop the antibodies against the protein of that bacteria or a virus, conjugate it, and do the immunohistochemistry. By this way, you are not only going to detect the etiological agent in the tissue, but you are exactly localizing the pathogen in the tissue, where it is, and how it is doing the pathology in the animals. That also can be studied with this immunohistochemistry. Usually, this is very widely used in the cancer, but today it is not our topic. Maybe Dr. Sood has already discussed about it. Then coming to the another latest technology is multi-TOF MS. It's a wonderful technique. This is very rapid, and it can distinguish between the various species, strains, and down to the lineages. There are lot many things that can be done. And it is very short turnaround time is just 10 minutes. And but the only thing is here that you need an isolate for this. It cannot be done on the clinical sample. So for doing this test and identifying the species or the strain, you need an isolate on the agar plate or whatever the plate is there. You need to isolate the organism, purify the organism, and then instead of going for biochemical and PCR, this multi-TOF MS can help you for the identification of the exactly species, strains, lineages. Everything can be done with this. It's a wonderful technique. Even we use it. We have in our college, and we use it. You have to just have the isolate and then do the multi-TOF MS. Even it can tell you the which if it is in the database. It will tell you even from which serogroup it is, which serotype it is. So that is the beauty of this multi-TOF MS. That was about proteomics, and this multi-TOF MS is again day by day they are going to improve. There are different companies which are coming with multi-TOF, and they use one company use the cell wall proteins for the bacteria to identify them. Some use ribosomal proteins to identify them. So like that, many companies, bioMérieux is there, Bruker is there. They have wonderful machines to identify this. So after proteomics, nowadays what is getting upper hand is the molecular test, DNA or RNA based test. Initially, they were used for the forensic purposes only, but looking towards their capability and the utility, now they have been used for the detection of the infectious agent in the clinical sample. Whatever the infectious agent and what sort it is immaterial. It should have RNA or DNA, that's all. And without RNA and DNA, the infectious agent is prion. Just one disease which I know, prion protein disease, scrappy. All others, they have either RNA or DNA in them, and you can use these molecular techniques for the identification of them. These techniques are initially it started with RFLP analysis, then STR analysis, AFLP analysis, mitochondrial analysis, it was, and PCR analysis. PCR has taken it way forward, and with the PCR, lot of developments has been taken place in this. So it is mostly wide, PCR analysis is mostly wide, and there are a lot of advantages of it. It is not only that the pathogenic organism is there in the tissue samples. Like I will give you one example of Escherichia coli in the gut. So there are pathogenic Escherichia coli in the gut. There are non-pathogenic or beneficial Escherichia coli are also there in the gut. So then how to differentiate them? If I have some samples, stool sample from a dog and suffering with diarrhea, and if I get a Escherichia coli in that, whether it's a common cell or it's a pathogenic which is causing disease. So this discrimination power is not available with the routine isolation and biochemical test. All will be same. That discrimination power the PCR has. So that can be done with this. Also the virus detection. Now, every lab is not handling, there are many labs working in India for COVID-19, and all labs are not handling infectious, isolating the virus and confirming it, which is the traditional way of diagnosis of viral diseases. But now they are using PCRs. So because of this PCR, I think there are maybe 200, 300 labs in India working and doing a daily 4 to 5 lakhs tests per day because of this PCR technology. Just imagine if PCR would have not been there, how could we have diagnosed these 4 lakhs cases per day? It was not possible to handle in a very specialized laboratory. So these are the advantages of PCR technology or molecular technology. Again, one advantage is that you can know the virus load by doing this PCR test. There are different types of PCR. Depending on your objective or your need, you can go for which kind of PCR can be used. Multiplex PCR, if you want to detect 3, 4, 5 diseases at a time, can go for multiplex. VNTR, within that bacteria, if you want to categorize or characterize them, you can use VNTR, nested PCR, qPCR, that is very widely used in the COVID diagnosis. Hot start here, touchdown. There are different types of PCR. These are very few. There is a long list. Maybe again you can double the list. So in COVID nowadays, all the labs they are using qPCR, using Tuckman technology. What are the advantages of this qPCR over the conventional PCR? In real-time PCR, you can detect the disease when the reaction is going on. But with the routine PCR, you have to run the reaction. Maybe it will take 2 hours, 30 minutes to 3 hours. Then you have to run the gel. And in the gel, if the PCR product is there, then you will confirm it. But that is not the case with the qPCR. When the test is running on the basis of the graph, you can say that the disease infective agent is present or not. And at each cycle, you can collect the data. That is the beauty of this. Also, it takes shorter time as compared to the traditional PCR. So these are few things. It is more sensitive and more specific than the traditional PCR. So that's why now in COVID-19 also, we use qPCR with the Tuckman technology, and that is helping the country a lot for the screening of the all samples at a time, many of the samples at a time. Then the development in this is LAMP, loop mediated amplification. This technology was invented by Japanese scientists in 2010. And in the routine PCR, we design a primers. When we design a primers, there are certain criteria to design a primer, that it should not form a loop. That is the main criteria. In the routine PCR, what we do, primer designing, it should not form the loop, and self amplification should not be there. That point has been well encased with the Japanese scientists to develop this LAMP assay. So they designed the primer in such a way that there will be formation of loop, and the PCR reaction will be continuous. PCR reaction will be continuous. So this is how the LAMP has been developed to know the etiological agent in the sample. This is like ELISA, we have put it in the reverse ELISA format or lateral flow format. This is the conversion of PCR, taking out from the PCR machine or qPCR machine out in the field, and you can do this LAMP at the bedside or on the farm. Just you need the water bath. Incubator water bath, you just need 60 degree, which can hold the tubes for 60 degree, and then you can do the testing. Here, the turbidity developed is the indication of the positive sample, and turbidity negative is there, sample is negative. This is very simple. And in the month of May, Honorable Minister Dr. Harshvardhan, he said that RT LAMP kit soon for the COVID-19 diagnosis. By this way, if we have these kits available, I think we can double the analysis. Like today we are doing every day 5 lakhs testing, even more. We can go up to 15 lakhs, 20 lakhs testing per day if the RT LAMP kit is available. It is cheaper as compared to the Tuckman PCR also. It is fast. It doesn't need a specific specialized equipment, very costly equipment, very easy to do. But the only thing in this LAMP is there are chances of false positivity, which needs to be rectified at the stage of primer designing itself. When you design a primer, that time if you take care, I think you can get rid of this false positivity and you can use this. Also, there is a latest development, but there is no kit in the market, so I didn't put it here. After LAMP, after doing the LAMP for just 10 minutes, this routine LAMP needs 30 minutes, 30 minutes test. But if you can go for the LAMP for 10 minutes and then run the solution in the lateral flow, and then you can diagnose the disease. In 2012, when I was in UK, we used to work on the brucellosis that time, but still it has not come in the market, so I didn't put in the presentation. But those techniques are also now coming. Just do the LAMP for 10 minutes on the clinical sample. That is very important. You can do it on the clinical samples and run this solution in the lateral flow. If you get the test line positive, your sample is positive for the disease. That is a technique which is coming out now. Then in development is DNA hybridization. Particularly this PCR and all those techniques, they are good for the diagnosis of the 1 or 2 or 3 or 4 or 5 diseases. But if you have a reproductive disorder in animal, and if I ask undergraduate student to enlist the diseases which causes abortion, he may write some 80 diseases in it. And I want to screen my farm, how many diseases are there that I need to take care in my farm. So in that case, you can go for DNA hybridization assay, microarray assay. Mostly this was used in the disease progression and the mostly for the how the transcription happens at the in the tissue level, or that was initially it was invented, but now you can use it for the different diagnosis of the diseases. You can use it. And latestly, see, these are the different 3 eras of the sequencing, I tell you. This is the routine sequencing method that used to be done, and we used to read on the X-ray film. And in this era, when one whole genome was thought, particularly human whole genome was thought to do the sequence, it took near about 5 years with 40 labs working globally to sequence one whole human genome, and lot of funds. So there are now developments. From here, we have moved to this type of sequencing, Sanger sequencing. It is bit faster, less cumbersome. This was very cumbersome, and this was less cumbersome. And now we have NGS. So with this, NGS has evolved. It has solved many our problems, and it has taken the diagnostic to the very higher level, to the one level above, and it has helped the diagnosis of the diseases, or particularly of the infectious diseases, very fast. So with this NGS, now we have one term which has come up, that is metagenomics. Metagenomics is the all genomes present in that sample. All the genome which are present in that sample, which includes maybe bacteria, virus, or protozoa, fungi, whatever it is there, we are getting it from that sample. And it is very well used nowadays for the clinical infectious disease diagnosis, and it is very one step closer to the reality. One case I am going to present it here. Nowadays, it is not the infectious agent which cause the disease. There is a one term which is coming, dysbacteriosis. It is the proportion of the various bacteria present in that organ. If it is a changed, their relative abundance is changed, there is a disease. And we did some work with the endometritis in cattle about the dysbacteriosis, and we have found that in infected animal, the certain kind of bacteria, they are on the lower side, and certain kind of bacteria, they are on the upper side. If we just correct this, their relative abundance, the animal will be again in stress and it will conceive. So this is wonderful work that we did, and this term is called as the dysbacteriosis. This dysbacteriosis leads to the disease. It is not the one single infective agent which is responsible for the disease, but just their relative abundance. If there is a change, there is a disease. And if we correct it, I think we can treat the patient accordingly and get the benefit out of it. Then there is one technique which is FISH, which is very recently been introduced. Last 15 years it has been introduced. Initially, it was used for the detecting the defects in the cytogenetic laboratories. The chromosomal abnormalities and all, there it was introduced. But looking toward the advantages of it, it has been used in the detection of the infective agents also in the samples. So this is like just a DNA hybridization which was done on the plate. Here we do it in the tissue itself, and it will help you in the detection of the etiological agent, maybe 1 or 2, 3 etiological agent, and localize them within the cell, within the cytoplasm, where it is, where exactly it is, and all that you can do with the FISH fluorescent in situ hybridization. This is also a wonderful technique, and you can detect the infective agent in the tissues. So for the molecular diagnostics, they are very expensive. Yes, I know they are very expensive. Particularly the metagenomic approach is very, very expensive. But still, I will be very strong supporter of it. Why I support it? Because It is not only detection, but there is a diagnosis of the dysbacteriosis and other conditions along with it. You can have the unculturable bacteria. See, with the routine technique, what is the first step is you have to isolate the bacteria on the culture, on the agar plate. But what about unculturable bacteria? There are certain infective agents which are not culturable. Still, we have not found any method to culture them. So these unculturable bacteria or the infective agents or the pathogens can be identified with this metagenomic approach, with the molecular diagnostic, not necessarily metagenomic, but you can use the simple PCR also to detect those unculturable organisms. There is we need a very rapid answer nowadays. Diagnostic techniques like when the samples are sent for the COVID-19 at 5 o'clock today in the afternoon, next day morning, 10 o'clock, the phone will ring. Sir, what is the result of sample? We don't have patience nowadays. We don't have patience nowadays. Think that with the earlier techniques of the virus isolation and confirmation, sample has coming to today, we have put it on the cell line and we have to give 5-6 blind passages and then see the CP and all, how much time is required. But now with the no patience in the human thinking, we need a rapid answer, and that can be given only with these molecular techniques. Also, these molecular techniques can help us to know the prognosis of the disease, or even it helps in the management of the disease. That is the advantage. There is you can do the quantification of the virus load or bacterial load in the clinical samples. If you have the paired samples, like one sample taken today on the day of admit and the sample taken 5th or 10th day of the day of admit, and if you know that virus load is coming down, you can tell that patient is going to recover. The animal is going to be recovered. And if the virus load is increasing, you can say that prognosis is grave, or don't know what can happen in such cases. So this is beauty of this molecular test, PCR, reverse transcriptase PCR, or qPCR, or even metagenomic with NGS. You can do this. Also, the researcher, it helps a lot, because with the isolation, doing biochemical gram staining, you don't know whether it's a mutant of the earlier bacteria or earlier virus is mutated or not. This mutation can be detected only with these molecular techniques and sequencing. And the era where we are living now, COVID-19, is highly mutating virus. 36 mutations is estimated in a year. So how to know this technique? How to know this mutation? These mutations can be known only if you go for the molecular test. Otherwise, you cannot. And if you know the mutations, what has happened, you can design the control strategy in that way. You can change the vaccination strategy in that way. You can change the vaccine strains in that way. Infectious bronchitis, which is also a coronavirus, has taught us a lot of lessons over the period of 80 years. We have developed some vaccine, it has evolved in a very different way. We have developed vaccine, it has evolved in a very different way. So all the evolutions we could check because of these molecular diagnostic techniques. So this is a beauty, and that's why I'm very strong supporter of these molecular techniques, though it is expensive, but we need them in the diagnostic of the infectious agent. Then there is one other technique, that is use of bacteriophages for the diseases, infectious diseases, particularly bacterial infections. You know, bacteriophages are the viruses of the bacteria. They are viruses of the bacteria. But we can take advantage of these bacteriophages in the early diagnosis of the diseases. Like one example I have given here about the Borrelia. If you design a PCR for the genome of the Borrelia, the sensitivity is quite less. But if you develop the PCR which detect the bacteriophages which are very specific to Borrelia, the sensitivity increases 6 times. 6 times is not a joke. So we can use these bacteriophages as a novel diagnostic. We can design our PCR in that way. Also, there is one company called bioMérieux, France-based company. they have tagged some fluorescent dye to these bacteriophages and use these bacteriophages for the identification of the bacteria in the sample. And this is how they are detecting. Even I tell you example of listeria, because we work in the listeria with the routine traditional techniques, isolation, PCR and all. We need at least minimum 2 days for the PCR confirmation, because we have to enrich first and then go for the DNA isolation and PCR. But with these use of bacteriophages, tagged bacteriophages and detection of salmonella or listeria in the meat sample, they have a technology to detect this within 24 hours or before that. So now this is such an era that I have a capacity to do it. Everyone has a capacity to do it. But who is the best? The person who will do it first is the best. So if you reduce the time of the detection of the infectious agent, that is the best. So bacteriophages has this. We can use these bacteriophages for the diagnosis of this infective agent in the clinical samples. Some of the bacteriophages, lytic, particularly lytic bacteriophages, can be used for therapeutic also, but that is not a today discussion, so I won't talk on it. But there are certain bacteriophages that we can use for the detection of the infectious agent, either by doing the PCR of those bacteriophages, or PCR targeting those genome of those bacteriophages, or labeling them and identifying in the clinical samples. Then since last decade, the biosensors are a lot of coming in discussion, and these biosensors can be used for the diagnosis of infectious agent in the clinical samples. And quantum dot is one of the technology. Basically, they are semiconductors, and they are nanomaterial semiconductors, and they can be explored for the detection or increase in the sensitivity and detection of the infectious agent, which is in the various less quantity that you can use this quantum dot. Then there is a FRET. This FRET technology also can be used. It uses the optical fibers, and this FRET is mostly used. Both of these, quantum dot and FRET is mostly used in the detection of the food pathogens. Detection of the food pathogens, particularly listeria or salmonella or like that. You can use this energy source material tagged with the antibodies, and as soon as they attach to the pathogen, the energy is released, fluorescence light is emitted, and that you can detect. So these biosensors, there are different types of biosensors. I have just put 2 or 3 here, but there are different biosensors that can be used. Depending on the need, you can use the different technology of the biosensors and detect it, detect the pathogen present in the clinical samples. Then there is one approach coming, syndromic infectious disease testing. Like I have given you one example of the endometritis, but there are a lot of syndromic diseases like diarrhea, respiratory affections, abortions, urinary affections. So there are many syndromic kind of things, and you have 10, 15 diseases or maybe 50 diseases on your list that you need to diagnose, you need to confirm from that sample. So in such syndromic diseases, there is one company called BioFire, and BioFire has come up with the technology that it is shown here, and you can have 100 diseases test at a time from the clinical sample. You have to isolate the DNA by the kit. I think now you can do within 1 hour or before 1 hour, and put it in the BioFire machine for the direct diseases. One of our collaborator, human collaborator, we have one L project, one of the human collaborator, they use this machine for the direct patients. So it is very fast. Within 2.5 hours, you get the diagnosis of all 50 diseases. It is very easy to do. You have to just isolate DNA with the kit and put it in the machine. The machine will do 2-3 PCR, and you give the it will give the result, and very comprehensive. I told you it can go to the extent of 100 pathogens in a sample. Whatever the technique they are using, they are presently limiting to 22 because there are 22. And beauty of this is whether it's a bacterial or viral or something, it can do same time. Like diarrhea causing bacteria are also there, diarrhea causing viruses are also there. So all can be done within this. So syndromic infectious disease testing is now coming up very fast, and BioFire has various machines for the various syndromes, and it's a very good technique. You just need a cassette and isolate the DNA and run it in the machine, and after 2.5 hours to 3 hours, you get the results. So now the era has come for the precision diagnosis. This is very important. Not only the fast, accurate, but precise diagnosis is very, very important. So with the routine technique, it takes near about days to months. The sample coming, putting on the plate, getting the isolate, confirm it, do the AST, and then give the treatment. It takes some days to months. But with this, the advantage of the NGS or these molecular techniques, I think these months has come to the hours. So now within 24 hours, you can have the disease diagnosis and the treatment is given. I will refer one very good article which is published in New England Journal of Medicine in 2014. And that article is of cerebral leptospirosis. And it was of the 14-year-old boy who was suffering with some cerebral symptoms. And all sort of investigations have been made, but nothing could be diagnosed. And he was not recovering. So that time this metagenome was just new, coming up technology. So one of the physicians, he removed the cerebrospinal fluid from that patient and did the metagenome. And to the surprise, it was a leptospirosis. And when the treatment was given, on the 7th day, the patient was back to home. So this is the precision, which was never thought just by doing the NGS of the samples, cerebrospinal fluid sample. They did the NGS and then diagnose and treat, and the patient was very recovered and went home very happily. So that was published in 2014, and after that, like a storm, you just name the case, and that case has been diagnosed by this technology. You name the bacteria, viruses, fungi, parasites even with this NGS technology, and this is revolutionized the field of the diagnosis of the infectious agent. So from 14 to now 20, you just name the disease, it has been diagnosed with the NGS from the clinical sample. So this is beauty, and this is the precision diagnosis will be required in future. At present, it is for the human science, but days are not far that within 2-3 years, we will also need it in our veterinary science. Then coming to some future techniques, that some biomarkers, particularly host protein response. After infection, what happens, how the host respond to the infection. And there are certain proteins of the pathogen also, which are only expressed when there is a disease, not with the routine their propagation. So these either host protein response or the pathogen protein response when there is a disease, these are now further explored for the diagnosis of the disease. Then there is one technique same as that of the NGS and application of NGS, genome detective. This is a sort of a, see, by doing NGS, you have a lot of data. Large amount of the data is available with you, but you should know how to perfectly identify in it. So this genome detective is the analytical tool from this data. It will help you to exactly pinpoint what is the disease and what is the relative abundance, whether it has reduced or increased or something like that. That can be done with this genome detectives. A lot of work is going on that. And one very coming up area is though it is not part of today's talk, but I thought AMR is a topic of hot cake now. Now the technique is coming, culture-free genome-based detection of the AMR. So for the detection of antimicrobial resistance or MDR pathogen, we have to isolate it and then do the testing, MIC testing, and then we know that this is resistant to these bacteria. But the technique is evolving now with the bioinformatics tools, culture-free genome-based detection of AMR. So if you have an organism, just sequence it and you will know for the which antibiotics it is resistant or which is sensitive. So just I have enlisted 3, but long list is there, which is coming forward for in this detection of the infective agent in this era, and this will continue. Maybe if we are able to diagnose the disease within 13 hours, 14 hours, the day will, some person will come, I want in 4 hours, 5 hours, like that, with the high precision. So this is a continuous evolving technique or technology, and a lot of techniques will come in this detection